Okay, hi everyone. My name is Min Tang, or you can call me Tommy. Now I'm a senior scientist in Shirley Liu's group at Dana-Farber Cancer Institute working on single-cell transcriptomics and epigenomics. My background is a hybrid of experimental biology training during my PhD at the University of Florida and the computational biology training during my postdoc in MD Anderson Cancer Center. So today I'm going to give you a lecture on single-cell ataxic. By the way, this is my Twitter handle and my blog address. So I mainly tweet and blog about the data analysis and sometimes complaints about bioinformatics. So if you're interested, feel free to follow me or check out my blog. Okay. We know DNA wraps on nucleosomes in eukaryotes, and transcription is tightly controlled by transcription factors. The accessibility of a chromatin region to a specific TF is critical in gene regulation. So ataxic or assay for transposes accessible chromatin with high throughput sequencing is a method for determining chromatin accessibility across the genome. So it utilizes this hyperactive TN5 transposes to cut the DNA which is open, and it insert sequencing adapters into the chromatin regions. Then high throughput sequencing yield those reads that indicate this region of increased accessibility. As you may know, there are many other methods to assay chromatin accessibility. For example, we have this mNA-seq or DNase-seq. For the mNA-seq, it uses this enzyme called microcalcanuclease, which preferentially cut the linker DNA. So the end result, what you get is the nucleosome positioning information like this. And the DNase-1 is used to preferentially cut the open chromatin. So this is the profile you get after DNase-1 sequencing. And for ataxic, it uses TN5, as I just mentioned. And TN5 cut both the open chromatin region and also the linker region. So in the end, you get all the accessible regions in the genome. So why ataxic is, oh, by the way, so because it cuts, the TN5 cuts both the linker region and also open chromatin region. So after you make a bulk, for example, for bulk ataxic, after you make a sequencing library, you can load the library into TapeStation and check the fragment length distribution. So you can do it in the lab, and you will find this pattern. So you should see a peak with smaller than 100 base pair, which indicates the open chromatin region. And then you will see a peak at 200 base pair, which corresponds to roughly the length of one nucleosome. And then 400, which corresponds to the two nucleosome lengths. And after you get the data, you can also do this computationally because you get that R1 read and R2 read, and then you know the length of that fragment, right? So you can make this histogram computationally, and you will see similar pattern, this periodicity pattern. So there are so many methods for assaying chromatin accessibility. So why ataxic is gaining popularity? So first, it finds all the accessible chromatin regions, i.e. all the transcription factor binding sites in the genome. I have a question. So are they all active sites or repressive sites? Those are open chromatin regions. Any answer for that? So the idea is the repressors can also bind to the chromatin and protect the DNA from being cut by a TNF5, right? So those accessible or open regions can be a repressive sites as well. In addition, it's really fast and easy and requires low starting material. In the early days, let's say, for example, ChIP-seq or other sequencing technology requires a lot of starting material. And for tumor samples or samples that are hard to get, it's not possible to do with other technologies. But with ataxic, you can start as low as, like, for example, 2,000 cells. That's why it's gaining popularity. And this is further developed into single cell resolution, which was commercially available from 10x Genomics. So you can buy these barcoded gel beads from the company, and you can make single cell suspension or single nucleus suspensions. And what you can do, you can run both of them through this 10x machine. At the right speed, you will see each gel bead will be associated with a single cell within a single oil droplet. And all the PCR reactions will occur within the single droplet. So all the open crumpled regions will be tagged by this same cell barcode for that cell, and which we can separate computationally later after we get the data. Okay, let's look a little bit closer to the library construction. So we know here each bead contains the P5 is the Illumina sequencing adapter, and this 10x cell barcode is 16 base pair, and this is the this sequence, this fragment sequence, it corresponds to the reverse complementary adapters TN5 inserted. So after the first PCR, so the DNA polymerase will extend the DNA from 5' to 3', right? So you extend from here, and then you extend from 5' to 3' in the reverse strand. So this is what you get. And then at the second round of PCR, you use another set of primers. So for example, now you use a P7 is also the Illumina sequencing adapter, and you followed by this sample index 6 base pair. And this is just reverse complementary to the other adapter that was inserted by the TN5. So after the PCR, okay, 5' to 3' fill in the gaps, and 5' to 3' fill in the gaps. And this is the final single cell ataxic library that you get. Okay, so in last two lectures, last two lectures, you have learned about single cell RNA sequencing. And let's just compare these two different libraries. So this above is the 10x single cell RNA-seq library. And here is the single cell ataxic library. So R1, the forward read for single cell RNA-seq contains only the 16 base pair cell barcode plus the unique molecular identifier, UMI. It's 10 base pair for version 2, and later for version 3, it becomes 12 base pair. So either the R1 read will be either 26 base pair or 28 base pair. And the R2 read contains the genomic information. And if you compare to the single cell ataxic library, the R1 read will contain the 16 base pair cell barcode, plus some genomic information there, and also the reverse strand, which also R2 read, which also contains the genomic information. So for single cell ataxic, it's a real pair end, but for single cell RNA-seq, actually you only get one single end because the forward read doesn't contain any genomic information. So I have a question here: why there's no UMI in single cell ataxic libraries? So this UMI in single cell RNA-seq is used to differentiate the PCR duplicates, right? Why we don't need that in UMI? UMI in the single cell ataxic library? Anyone has an answer? Nope.
Because we only have 2 copies of each chromosome.
Right, so we only have 2 copies. So anything more than 2 copies, we know it's a duplicate, so we can computationally remove, flag those fragments. Okay. Other methods, in addition to the droplet-based, are being developed. So similar to like single cell RNA-seq, there's the plate-based method in which a single cell is sorted into a single well, like for example, into 96 well plate. There's also this split and pool strategy also initially was used for single cell RNA-seq. So each So you first split the cells into different plates with different barcodes, and you collect them, pull them again, and then split again. So each cell will undergo a distinct path as marked by the barcode added at each step. But the problem is that you need a lot of cells to start with, because each round you can lose some of the cells and it's not commercially available. So it could be like labor intensive. So far, 10x genomics is still the most popular method, because you buy the reagents and the machine, you run through the 10x machine, you're likely to get some good quality data. Of course, it's a little bit more expensive, so but you need to kind of balance. Yeah. Right. So just in the very early days, just for the purpose of developing the technology, people only do single cell ATAC-seq alone. But the epigenetics should be interpreted in the context of gene expression. People usually are more interested in doing single cell RNA-seq and the single cell ATAC-seq in the same tissue or the same sample. So one can split the same sample into two tubes, and one undergo single cell RNA-seq assay, and the other undergo single cell ATAC-seq assay. So somehow because they are from the same sample, not exactly the same cell, but still you may be able to map those two data sets together. But early this year, 10x released a commercial solution for single cell RNA-seq and a single cell ATAC-seq on the same cell. They call it multi-ohm data, which is also gaining popularity. And we will analyze this multi-ohm data in homework 5. Okay. Yeah. So I will do a first Zoom poll now, and I will stop. Okay, let's continue. Okay, before I go to data analysis for single cell ATAC-seq, I want to emphasize the importance of experimental design to avoid batch and confounding effects. This is the same for any experiments. In previous homework, you worked on a batch effect correction problem for bulk RNA-seq data. And Martin, your TA, also includes this slide in the single cell RNA-seq lab. Before you start any experiment, always involve a statistician. This will help you to avoid such problems. As the statistician Ronald Fisher said, to consult the statistician after an experiment is finished is often merely to ask him to conduct a postmortem examination. He can perhaps say what the experiment died off. Right. So really you should have a balanced experimental design, and also can mix different groups of comparisons that you want to make, mix them together for different batches. So after, say, for example, after you obtain the data and do a PCA, you know this is good, because the same kind of samples that cluster together, even they're from different batches. For single cell studies, one can do multiplex and barcode before different samples, and then run them together within a single 10x controller, and that can help to mitigate this batch effect. Before I go to single cell ATAC-seq analysis, I want to just summarize the single cell RNA-seq analysis. This is a typical workflow. In the single cell RNA sequencing lectures, you also see this slide. So we usually get the sequencing data off the sequencer, and then align them to the transcriptome and do quality control, normalize the data somehow, and then do dimension reduction, whether it's like PCA, t-SNE, UMAP. And then cluster cells and do cluster annotation. And there's some downstream analysis that you can do, trajectory inference, differential expression, and compositional analysis, etc., etc. The single cell ATAC-seq analysis follows a similar workflow with some special treatment for the count metrics, since the data are much sparser than single-cell RNA-seq. So as you can see, there are many steps in the workflow. And to streamline the analysis, our lab, Shelley's group, developed a tool called Maestro. And to address these challenges of processing single-cell data, we developed this tool, Maestro. Maestro is an integrative analysis workflow based on SnakeMake, and for both single-cell RNA-seq and single-cell ATAC-seq data analysis. So you're going to use this tool for your homework 5. I believe some of the students may have already tried it. If you are not familiar with SnakeMake, I would suggest you to look it up on the internet. It was developed by Johanna also, who was in Shelley's group as well. And this SnakeMake is gaining popularity in the bioinformatics field for writing workflows, and it's Python based. So if you know Python, you can learn it easily. Okay. So it's a SnakeMake pipeline with steps ranging from pre-processing from FASTQ files and do some quality controls, QC, and downstream analysis, including clustering, differential gene expression or differential peak enrichment, cell type and annotation, and the regulator identification. So it also supports integrated analysis for both single-cell RNA-seq and single-cell ATAC-seq data if you have samples run through both assays, or you can also handle the multi-omic data as well. So I will explain the features in the following slides in a little bit more detail. So, okay, the first step when you get the data, it's always align the data into the reference, whether it's a transcriptome or genome, right? For CellRanger is a 10x genomics solution, so it's from the company. It can do the single-cell RNA-seq analysis, also single-cell ATAC-seq data analysis. The problem of that is it's kind of slow. It may take maybe over like 20 hours to just process a medium-sized data. And for RNA-seq, actually we use this tool called StarSolo. It's much faster, and it's 10 times faster than CellRanger. So we use this tool in Maestro. And StarSolo was developed by the same author of STAR. I believe you used STAR for bulk RNA-seq analysis in your previous homework. For ATAC-seq, there are different aligners that you can use. A BWA, which is popular, was developed by Han Li, and Minimap2 is also developed by Han Li in our department. It's 15 times faster than CellRanger. And I believe you also, in the previous lectures, you also use this called pseudo alignment tool to do quantification for RNA-seq data. The question here is that can you use Salmon or Callisto, those pseudo alignment tools, for single-cell ATAC-seq data? Can you use that? So think about the difference between ATAC-seq and the RNA-seq. Right. For RNA-seq, you know the features that you're looking at, right? So for example, in human we have 20,000 genes. You can count how many reads aligned into each gene. But for single-cell ATAC-seq, you don't know what to start with. You want to call-peek and then find those open chromatin regions, then count number of reads in each peak. But actually, you can also do that if you have a public available data set which combines all the DNAS1 or bulk ATAC-seq open chromatin regions. You can consolidate them into a big, more like a reference. So you can count number of reads in those potentially open chromatin regions. And using that, you are still able to do like clustering, find new clusters. But again, any reads that not aligned into those public available potential open chromatin regions will be missed. So you can still use it, but then you lose some information. And of course, sequencing has errors. Right? You will need always to do barcode correction. And for 10x, they have their they call it whitelist. So they know the exact sequence they put into the gel beads. And after sequencing, you can compare your sequencing results, the cell barcode, out from the sequencer to the whitelist, and then you can correct based on the Hamming distances. Okay, for bulk level sample QC, that we always call peaks first using Max2, and this is by collapsing all the cells together as a pseudo bulk, and then you can call peaks using Max2, which is a popular algorithm for ChIP-seq data. And it was also developed in Shirley's group. And for bulk RNA-seq, we use RCQC and I realized they actually just recently published RCQC2 for faster quality control for RNA-seq data. And there are some other standard ChIP-seq QC metrics for ATAC-seq, for example, the number of high quality peaks, fraction of reads in peaks or promoters at the bulk level. And an example here is that so you can do a bulk cell ATAC-seq, and also you can do single cell ATAC-seq side by side, and you will see you can kind of collapse. So here, each row here is a single cell, and each black bar here is one read. So if you kind of aggregate those reads together, it should give you a similar profile, just like doing a bulk cell ATAC-seq. So for technology development, quality control, one can compare the consistency between replicate runs, right? For example, here, for this particular cell line, GM12878, you can run 2 times, so one batch with 200, around 300 cells, the other batch with 4,500 cells. And then here, each dot here is the peak location. So, so you can plot a correlation to see log2 transform the count per million to see how well those 2 batches are correlated. So a successful experiment will have a high correlation. This is another Baryon experiment in which we have mixed mouse and human cells, just like in single cell RNA-seq, as introduced by the last lectures. So 820 is a mouse cell line, and GM12878 is a human cell line. So ideally you expect, okay, those cells are only from the mouse cells, and those cells are only from the human cell line. And for those dots, those are doublets, which contains reads mapped to both human and mice. And as expected, if you overload the 10x machine, you start to get more doublets. So the number of cells per droplets actually follow a Poisson distribution. To minimize the number of cell doublets, the single cell suspension is loaded into the microfluidic device at a very low concentration, so making it very unlikely to have 2 cells enter into the same droplet. But one thing is that most of the droplets are fully functional. They contain the barcode is the beads and also the reagents, but they do not receive a cell, and therefore they are wasted. So there are other methods, for example, cell hashing by labeling the cells from different samples with a barcode first, and you can pull cells together and overload the 10x machine. I think the recommended maximum number that you can load into a single 10x lane is like 10,000 cells. But with this cell hashing or pre-barcoding strategy, you can overload the cells. You can overload the cells into the machine. And however, this is not necessarily used in the common experiments. This is really just for technology development. Okay. Now we go to the real QC for each cell. And we know most of the promoters are open, and we should have enrichment of ataxic signal in the promoters. Right, so you can either have percentage of reads mapped in the promoters or percentage of reads in the peaks. The higher the better for a good experiment. And if you, for example, if a higher percentage of reads mapped to mitochondria, just like in single cell RNA-seq, you know that's not good. But the exactly cutoff, like for example, the mitochondrial rate need to be determined by each experiment. So each experiment may have different level of mitochondrial contamination. So for here, this is the Q for this quality control here. Like for the x-axis, this is the total number of unique fragments in each cell. So here, each dot here is one cell, and the y-axis is the TSS enrichment score. So you can this TSS enrichment score is was defined by ENCODE for bulk RNA-seq, for bulk ataxic, and here we just borrow it for single cell ataxic. And if you really want to know the details how to calculate this TSS score, so you can go to this link here. I wrote a blog post about how to calculate this. And so in this example, you see, okay, people usually use like 1,000 fragments, unique fragments as a cutoff. And here they use, I believe it's TSS score of 8 as a cutoff. So those are the high quality cells. This is for both human and mouse, so similar. And in Maestro, we actually use this more simpler metric. It's called fraction of reads in peaks. So, and essentially we get the similar pattern. So those blue cells are high quality cells with high number of uniquely fragments in each cell here, and also high fraction of reads in the peaks. Although it seems a lot, like those cells are a lot, but those cells are low quality and they contain very little number of reads. So you still get a good number of reads actually mapped to those good cells. And if you do a pseudo bulk again, using only those cells, it will have a better, like a fraction of reads in peaks after this cell quality control. Okay. So what else could go wrong? So I mentioned that there could be a doublet problem. So more than 2 cells, they're in the same droplet, although they are 2 distinct cells, but they receive the same cell barcode. And after sequencing it, we can't differentiate whether they're from same cell or 2 cells. And the other problem is that different barcodes actually can be from a single droplet. This is a paper published in 2019 by Caleb, who was our previous TA in last year. And here, within the single droplet, it could be you have 2, you can have like 2 beads which contain different cell barcode. And or you can have a single bead, but within that single bead, you have 2 different barcodes. So roughly like 5% of the beads contains multiple barcodes, and 4% of the droplet contains multiple beads. And this is only happens for single cell ataxic, but not for single cell RNA-seq. This is the 10x solution. And then this brought attention for them, and they later fixed this problem. So the idea is that you don't trust the data other people give you, or also you don't trust the specification that whatever the commercial solution tell you. But you really need to do a lot of sanity check or investigation, some detective work. Okay, this is another Zoom 4. So the question here is, which one is better for quality control for single cell ataxic? One is fraction of reads in peaks. Second is fraction of reads in promoters. Any answer? Which one is better? Or maybe there's no better or worse. Let me see the pool. Okay. And personally, I think the fraction of width in the promoters will be better than the fraction of width in peaks, because it really depends. For the first metric, it really depends how many peaks you have, right? Because the more peaks you have, the higher fraction of width will be in the peaks, and the number of peaks in a pseudo bulk, that can vary a lot by tuning the parameters, right? If you lose the stringency a little bit, you can get a lot more peaks, and then you get a higher fraction of width in peaks. But fraction of width in promoters are relatively constant, right? They're there. So I tend to use fraction of width in promoters. So I vote for that. Okay. The next step is to generate a count matrix. That's the key step in single-cell ataxic analysis. So as I mentioned, usually the FASTQ files are aligned to BAM file for each cell. And in the BAM file, there's a tag. If you look at the BAM file specifically, there's a tag called CB, or cell barcode, containing the cell barcode information, telling you which cell this particular fragment is from. So we then call peaks for the pseudo bulk. So aggregate all the cells together and call peaks using max2. And then we count how many reads in each of the peak for each of the cell. So in the end, you get this peak by cell count matrix. And this matrix is very sparse, and even much sparser than single cell RNA-seq. So as we mentioned, that's because we only have two copies of DNA, right? And for RNA-seq, you can have multiple copies of that same transcripts. So this matrix is quite sparse. Okay, there are some technical considerations for single-cell ataxic analysis. So one can either look at the reads on peaks in each cell. So this, what I just described, is peak-based. The other tool called Snap Ataxic uses a beam-based method. So essentially, it tiled the beams across the genome, like 5 kb beams, and then you count reads in this 5 kb beam. But we know like we have like 3 billion bases, and if you divide it by 5 kb, it will be around 0.6 million beams. So that's a lot of things, and the counting, the calculation may be a little bit more computation intensive. Then after we get the counts, we generate this peak by cell count matrix, or this beam by cell count matrix. As I mentioned, this matrix is very sparse. There's no UMI. And you can, because it's really sparse, anything bigger than 2, bigger than 1, you can assign if it's 1, and if it's 0, let's say it's 0. So make the matrix binary. But for this binary matrix, it needs a little bit of special treatment. We call TF-IDF transformation. So I will explain this in the next slides. So the downstream analysis starts from this sparse matrix. Let me explain what is TF-IDF. So this TF-IDF, or frequency inverse document frequency transformation, this was firstly used in this paper, Darren et al. 2018 in Cell. And they used this TF-IDF transformation to normalize somehow the single-cell ataxic data. And in the method section, it says, so we first weight all the sites for individual cells by the total number of sites accessible in that cell, and that's the term frequency. And we then multiply this weighted value by log, then parentheses 1 plus the inverse frequency of each cell across all cells. And this is the inverse document frequency. Then they use single value decomposition, SVD, or PCA on the TF-IDF transform the matrix to generate this low dimensional representation of the data. So if you really translate into R code, it's not that complicated. So if you use R a little bit, you can read code here. So you get the column sums of that matrix, essentially the total number of accessible region peaks, or the total number of peaks. And here, then you can just divide it. For each cell, you can divide this total number of peaks for each entry, right? And then now here you calculate the total the row sums, essentially how many cells for that particular peak has this peak or has this peak open, and this is this is just total number of cells in that matrix. And this is the inverse ratio, right? And now after that you just do a matrix multiplication. Oh, by the way, this function is implemented in Seurat, so you don't have to do it like manually here, like what I did here. But I do have a blog post here, link here to explain what exactly TF-IDF is doing here. And it is frequently used in text mining because that's why you see the term frequency or document frequency, because one can count the frequency of a word in a document and then get the word by documents matrix. It is also very sparse. And with this matrix, one can find which documents are more similar to each other. Maybe some of them are written by Shakespeare and then others are written by someone else, and everyone has different preference of word usage. Right. Okay, so after you get this TF-IDF transformed matrix, now you will do a dimension reduction. So you run PCA first, and then use top 50 components and build a k nearest neighbor graph. And this is the most popular or better performed method currently. And so you can then use this community detection algorithm to detect how many clusters in this KNN graph. And later you can visualize the clusters in either UMAP or t-SNE like this. However, I want to add one more note for the clustering, is because there are many parameters that you can fine tune. So for example, for the KNN, you can specify different K. In Seurat, it's K like defaultly is 30, but if you change that to 10, 20, it will give you more clusters. And there's another really critical parameter is called the resolution. So the higher resolution that you give into the algorithm, the more granularity that you get. So you also get more number of clusters. So the bottom line is that we can always subcluster the cells and get more clusters. And theoretically, each cell can be its own cluster, right? And it really depends on how similar you want the cells to be within the same cluster. So the function of mathematically identified clusters here need to be interpreted in the context of biology knowledge. So I would say the knowledge of biology plays a more important role here. So in other words, finding the clusters is math, but interpreting the clusters is science. Okay, so this is an example of single cell ataxic UMAP with and or without TF-IDF in transformation. So you're going to do an exercise in homework 5 as well. So the figure on the left is the raw matrix, but without TF-IDF, and then you do the cluster dimension reduction and clustering and project them in UMAP. And here on the graph on the right, we use this latent semantic index. Essentially, LSI is equal to TF-IDF followed by SVD or PCA. Singular value decomposition is the underlying method in PCA. So you will see if you, without TF-IDF transformation, the look of this t-SNE plot, this UMAP plot looks a bit funky. So you see this kind of strange patterns of those things. But after TF-IDF transformation, you see much better representation. So It's quite similar to the single cell RNA-seq cluster. Okay, after we assign the cells to each cluster, for example here, so we assign each cells into each cluster, one can optionally do a peak calling again. So why we want to do that, right? Because sometimes, remember, we call the peaks using all the cells, right? All the cells together and using max2. And if you look at here, there are some clusters they're relatively small, and the peaks which specific in those small clusters may be missed when you call peaks with all the cells together, because it just get diluted, right? So what you can do, you can call peaks again just within each cluster, and now you can merge all the peaks together and regenerate this count matrix. So now you will have more peaks than previous that you have, because now you have a lot more cluster-specific peaks. And this is very important for finding differential peaks, which is the next step. For example, if you don't start recall the peaks within each cluster, you're going to miss those peaks that are specific to those small clusters. And if you don't have it to begin with, how can you find them after a differential peak calling, right? You can't. So for the idea of a differential peak calling is to, for each row or each peak, you calculate a differential enrichment between cluster 1 versus all other clusters. So whether this peak is preferentially open or not. And similar to the non-parametric test method for single cell RNA-seq, you can use Mann-Whitney test or Wilcox rank sum test on the peak cell count matrix. And we know because the matrix is really sparse and 0 and 1s, and to overcome this, because if you have too many ties, Wilcox rank sum test doesn't perform well. So you can somehow normalize the data in each cell and then scale to 10,000 reads per cell. Or you can use that TF-IDF transformed matrix for Wilcox rank sum test. And there is a tool called Presto. It implemented a fast Wilcoxon test, which is 1,000 times faster than in Thread. Because you can think about, for single cell RNA-seq, you have 20,000 rows. But for single cell ATAC-seq, you can easily get 300K rows. So if you have used the Thread find markers function, it sometimes takes a very long time. And especially for single cell ATAC-seq, you might want to adopt this method because it will get you the results in maybe within 20 seconds instead of hours. So I recommend you check this one out. Okay. People are more interested in finding out what the transcript factors binds to each cell, right? And CROMWA was developed for this purpose. So what CROMWA does is for each cell, it counts the motif occurrence in the peaks, and one can derive a TF binding score for each cell. And this gives you a general idea of the TF activity in each cell. And then you can overlay the TF activity score onto your map. And you may find some clusters that are enriched by certain TFs. So for example, here is CBP alpha. Then you say it's kind of higher in this cluster and a little bit here. And ZEP1 is another transcription factor, and it's kind of the activity is higher in the other clusters. But we all know that the occurrence of a TF in a genomic region does not mean the TF binds there, right? So we can to do that, we can use public data sets to partially address the problem of annotating TFs. So in our lab, we collected 60,000 ChIP-seq, DNA-seq profiles from public deposit data set into a database called CISTRON-DB. So we collected the over 60,000 ChIP-seq data and DNA profile from public deposit data sets into Citron DB. And we also have this toolkit called Citron Toolkit to do some analysis with the public data. For example, if you input a gene, and it can tell you which TF binds to that gene. And if an genomic interval, it will tell you what transcription factors may bind to your interval here. And lastly, if you input the PIC set, it will output what factors from the public data sets have a significant binding overlap with your PIC set. And this is implemented by our tool called Giggle. So Giggle is doing something like BAT-tools intersect if you are familiar with BAT-tools, but it's much faster when the data are big. We can apply it to single-cell ataxic differential peaks in a cluster. So for example, after you do differential peaks for each cluster, you get a differential peak set. And now you can use the Citron DB toolkit to find which transcription factors may bind to those peaks. Right. For example, in this example here, so the x-axis is the different transcription factors, and the y-axis is the peak set overlap score here. So, and each dot here is different experiments, because for the same transcription factor, there could be multiple labs generated the same TF ChIP-seq. And also, in addition, many TF families, for example, STAT, you can have the STAT 1, 2, 3, and they have all similar binding motif and peaks, and they can overlap with each other. So to tease out which TF is functioning in a cluster, we can also check the expression level for that particular TF. And you want that transcription factor to have high expression as well as high overlapping score. Okay, this is another Zoom pool here. So first question is, what are the challenges with this single cell ataxic count matrix? One is it's very sparse. Second is there's no UMI. Which one? Okay, I see some people answered. Yeah, I think the challenge here is really sparse. As I showed you, it's sparser than single cell RNA-seq matrix. There's no UMI, but that's fine because we can computationally flag the reads that the PCR duplicates. And the second question is, what could be the issues using the peaks called from all cells? Number one is peaks in a rare cell type can be missed. Number two is the clustering will miss the rare population. Which one do you think is the issue using peaks called from the pseudobulk with all the cells? Okay. Yeah, the answer is number one, because the rare cell type can get diluted when you call peaks from all the cells. But very interesting, the clustering will not miss those smaller clusters, as I showed you before in one of the UMAP. Those clusters, they still get captured. Even some rare peaks are missed, because there's still a small percentage of those peaks will be called, and that is enough to separate them from other clusters. But if you really want to get the differential peak or this rare cluster specific peaks, you want to call peaks again within each cluster. And the next step is to do cell type annotation. Cell type annotation is still an unsolved problem. And the studies have shown like single-cell RNA-seq is still the best to define relevant cell types. It's simply because we know which genes are specifying which cell type, right? But how to annotate cell types from single-cell ataxic data is even harder, because with single-cell ataxic data, what we get is the peak by cell matrix. And to interpret the epigenome, as I said, it should be in the context of the gene expression level. So in order to transfer the single-cell RNA-seq information to single-cell ataxic data, one needs to convert the peak by cell matrix to a gene by cell matrix, just like single-cell RNA-seq. So in other words, how to approximate RNA expression level by the chromatin accessibility. How to do it? It turns out to be a hard problem too, because we know promoter openness is not a good proxy for gene expression. For example, in stem cells, promoters can open and poised to express, but not undergo a productive transcription. And RNA polymerase II can bind to the promoter, but store there and do not undergo productive elongation. And in previous ChIP-seq lecture, I believe you also learned the bivalent promoters in embryonic stem cells. So those promoters are poised to be active, but they're not active. But on the other hand, the distal enhancers can have also a big effect on the gene regulation. For example, the well-known sonic hedgehog enhancer is located 2 megabase away. So how to do that? So in Maestro, we use this. We implemented this regulatory potential score to model the gene activity score from the peaks. So it uses an exponential decay model. So first we can define the decay distance, for example, 10 kb here. And for each gene, you can check the surrounding peaks. If it is located at this peak is located at the TSS, you give the regulatory potential score of 1. So if a peak is 10 kb far away from the TSS, and the regulatory potential will be 1/2, because this is the half decay distance. If it's 20 kb away, it will be 1/4. So what we do is to sum up all the peaks nearby and as the regulatory potential score for that gene. So all models are wrong, but some are useful. So this is the simple model that we use to model the gene activity score from single cell ataxic data. And after we convert this peak by cell count matrix to a gene by cell expression matrix, just similar to single cell RNA-seq, using a regulatory potential score. So now you're ready to use CCA or canonical correlation analysis to combine these 2 metrics together. So CCA is a method similar to PCA. It was developed like in the 1930s to search for associations between 2 sets of continuous variables X and Y. So its goal is to find a linear projection of the first set of variable that maximally correlate with a linear projection of the second set of variable. So we can think of CCA as a generalization of PCA, where the variance we maximize is the covariance between the 2 matrices. And this is in Surratt. It performs a canonical correlation analysis followed by this L2 norm of the canonical correlation vectors. And then they project the data sets into a subspace defined by the shared correlation across different data sets. And then in the shared space, it identifies pair of the mutual nearest neighbor across the reference and the query cells. And this should represent the cells in a shared biological state across different data sets. In our case, it's single cell RNA and single cell ATAC. It can be also, a reference will be a single cell RNA-seq, but the query can be also a single cell RNA-seq. Let's say if you have a bigger reference with annotations, you want to annotate your smaller set of single-cell RNA-seq data set. Okay. Yeah, I believe in the previous machine learning lectures, you learned what is L2 norm and L1 norm, right? L2 norm is used in the ridge regression, and L1 norm is used in the lasso regression. So we compared the Maestro with other 3 methods to model the gene activity score. So for example, SURAT, it also has its own implementation of a gene activity score. It simply counts 2 kb upstream and 2 kb downstream of the gene body, and just sum up the ataxic counts and use that to approximate the gene activity. And SNAP ataxic calculates the gene accessibility only at the variable genes identified using single-cell RNA-seq dataset. And Cicero is another tool. It uses the co-accessibility between promoters and peaks. And we evaluated consistency between the gene regulatory activity from single-cell ataxic after label transferring and the gene expression level from single-cell RNA-seq in the same cell type. So showing here. So as you can see, the correlation is not that great, and that's kind of expected. This single cell levels are roughly like, but Maestro is kind of outperform other methods for all the clusters. And we also found, and after you perform CCA to do the label transferring, so each cell will get a prediction score just to say how confident this label transferring is. And we also found that the integration using our Maestro regulatory potential model has overall higher prediction score. Okay. And since the 10x released this multi-ohm data, which simultaneously profile single-cell RNA-seq and single-cell ataxic within the same cell, so we can use multi-ohm data to evaluate the label transfer accuracy. This is the last exercise in homework 5. So for example, here, for multi-ohm data, because we know the single-cell RNA and the single-cell ataxic are performed on the same cell, right? So for single-cell RNA, so you can cluster all the cells and annotate them, okay, whether they are CD4 cells, CD8 cells, or NK cells. And for single-cell ataxic, it undergoes its own analysis, okay, TF-IDF, PCA, then clustering, and then you can do label transferring, as I just mentioned. So you can borrow the cluster label from single-cell RNA to single-cell atac. So this is a label transfer. And for example, if you, because we know they are from the same cell, and you can check whether the annotation is correct or not. So for example, here, this single cell is annotated as CD4 in single cell RNA, also CD4 in single cell atac. So this is correct. But for cell 3 in single cell RNA is annotated as CD4 cells, but in single cell ataxic, it's annotated as CD8 cells. So this is incorrect. So what you can do, you can just calculate the accuracy, the overall accuracy of the prediction, right? And of course, you can also further separate them by clusters. So here is the cell type annotation from RNA, and this is the label transfer, the annotation from your ataxic data. And usually those like distinct cell types, they have a really good label transferring. For example, B cells, NK cells, they are quite distinct, and PDCs, so they have a relatively good label transfer accuracy. And as I mentioned, there's a prediction score for each cell. And if you plot them as density, so for those correctly labeled transfer the cells, you can plot those distribution score. And for those incorrectly labeled transfer cells, you can also plot the density for the prediction score. And you see, in general, those incorrectly labeled cells have lower prediction score. Right? This is the last question for your homework 5. Okay, one of the unique functions of Maestro is the ability to identify important transcription regulators for both single-cell RNA-seq and single-cell ATAC-seq. I believe you all used LISA before in your previous homework. So we can use LISA to identify TFs that regulate a list of given genes by using public data set we collected in SysRunDB. And LISA builds an epigenetic model based on a list of cluster-specific genes and find factors whose binding sites are most likely to regulate these regions. Right. So now for each cluster, you can have a cluster-specific gene, and you can just input that gene list into LISA. And to predict the driver regulators from single-cell ATAC-seq data, as I mentioned, Maestro can identify the transcription regulators using public available ChIP-seq data that are enriched in the cluster-specific ATAC-seq, single-cell ATAC-seq peaks. So you may notice that for single-cell RNA-seq, you can directly color the transcription factors with its expression level. So you want those transcription factors to have a high expression level as well, in addition to higher overlapping of that public data sets. And for single-cell ATAC-seq, because we don't have the expression data, and we can use the regulatory potential score to approximate the gene activity, the gene expression level of that transcription factor. And in this example, Maestro identifies, let's say, SPI1 or PU.1C, BP alpha and beta, and FLI1 as the top enriched regulators in the PBMC CD14 monocytes, and in both the single-cell RNA-seq data and the single-cell ATAC-seq data. So that shows at least is consistent from two different data sets of the same cell type. They are highly concordant, which gives you some confidence of the prediction. Okay. And this is a summary of single-cell ATAC-seq analysis. So first, we need to do read mapping using whatever the best aligner that you want. We use Minimap2 in Maestro, and then do a peak calling after aggregating all the cells together. And we also want to do a quality control at the sample level or at the cell level. Then we generate this peak by cell count matrix, which is really sparse and give us some challenges. We can then do a clustering, or dimension reduction, clustering, and visualization. And with after you cluster cells in different clusters, and you want to do differential peaks and annotate them each cluster with a master transcription factor. Also, we can use this regulatory potential score as a proxy for the gene expression level, so that you can integrate the single cell ATAC-seq data and single cell RNA-seq data. And you can label transfer the cell type ID from the single cell RNA-seq data to the single cell ATAC-seq data. Of course, you can also identify driver transcription factors in each TF using GIGO. I would say both technologies and computational methods are still fast evolving. There are so many exciting developments that are going on. So in the last slide, I just want to summarize some other single cell related areas that are under fast development. So for now, we only covered single cell RNA-seq and single cell ATAC-seq, but there are other single cell assays that is available now. So for example, you can do single cell ChIP-seq. You can do a single cell like DNA methylation or single cell DNA sequencing to co-mutations. You can also do site-seq to interrogate the protein cell surface proteins. And what's really exciting is that now you can do this multi-omics thing, right? For example, I just mentioned for the multi-omic data, we can simultaneously profile single-cell RNA and single-cell ATAC within the same cell. And there are even assays to do single-cell RNA, single-cell ataxic, and single-cell DNA methylation within the same cell. But of course, the caveat is that when you have multiple assays carried on within a single cell, the data quality may not as good. So there's just some caveats. So, and only when you play with data, you know how good the quality of the data are. And the other really interesting thing or advance of the technology is called spatial transcriptomics, which was highlighted as the year of the technology in 2020 in Nature Biotech. And there are many methods that are available now. So for example, Merfish, this is developed at Harvard in Xiaowei Zhuang's lab, and my previous advisor, Catherine Dulac, also worked with them using this method to profile mouse brain to classify the cell types in the mouse brain. And what this technology is cool is because not only you get the gene expression level, you also get the spatial information of those cells. And we know the spatial information of each cell can play an important role as well. And also, this single cell DNA sequencing data can be used to trace data. For example, in cancer research, you want to trace the clone mutation just based on mutation, then you trace the clonal evolution. But that can be really challenging too, because you only have a single cell, you only have like 2 copies of DNA, right? Just like single cell ataxic. And the calling mutation could be really challenging because you need to differentiate whether it's a real mutation or it's just a sequencing error, right? So with all those available tools, assays, single cell related assays available, we can do many things. We can define cell types, and we can define the trajectory. And now we also have the spatial information. We can reconstruct the spatial information in the original tissue. And those are really exciting. So we can also study different cell state. As I mentioned, the cell types may not necessarily be just discrete cell types like here. It could be just continuous phenotype. So it can. So the cells, we call it as a cell state, not necessarily cell type. So yeah, this is a single cell still is a really exciting field. If you have a chance, I would encourage you to study. Okay, finally, I want to thank Shirley for your invitation and your guidance, and Gali Bai, who is the associate computational biology, who made homework 5. It took us a lot of effort to make it. So I hope you enjoyed homework 5 and don't complain a lot. And also other co-authors for the Maestrom paper, and Luca, he's also a professor at Harvard. And we cited some of their papers. And I have some reference for just bulk ataxic and single cell ataxic here in the last slides. And if you want to read more, you can go and read more. And this concludes my presentation today. Thank you very much.
Thank you.